Okay, so let's talk about organization. How are things organized? Why are things organized? Why should we organize this stuff? Well, there's about 1.8 million species of things, living things on the world. We talk about plants and animals and bacteria and so forth. 100,000 species of fungi alone. Nearly 300,000 species of plants. 57,000 species of animals with backbones, called vertebrates. So fish, birds, mammals, reptiles, and amphibians. There's about 1 million species of insects, 1 million species. Thousands of single-cell organisms like bacteria. So even today, we have researchers that are discovering new things every year. So we have an estimation of 10 to 100 million species on Earth. We still haven't discovered a lot of stuff. So we have to attempt some way to make sense of this huge variety. So we have to organize things. Whether it's a bird, a mammal, bacteria, a plant. We've got to organize this so we know what we're talking about. Did we discover something new, or was it already discovered? So we have 3 domains of life. There's one called bacteria, something called archaea, which is basically this, well, I'll talk about it, and then eukarya. Eukarya breaks down into kingdoms, and we have 4 kingdoms that are in there. Protista, Animalia, fungi, and plantae. Basically, the 3 domains here, bacteria, archaea, and eukarya, bacteria are all single-cell organisms but have no nucleus. This is where your bacteria fall. Archaea are similar to this. They're one-cell organisms also. They don't have a nucleus, but they can live in extreme conditions. We won't go into details about it, but they are types of bacteria that, under hot conditions, these will just get destroyed. Under hot conditions, these will survive. They've adapted. This is much better than this one over here. It's getting better. Eukarya, that's where we fall in, animals, fungi, plants, and protista. These are one-celled organisms, but they do have a nucleus in there. So we got to decide, do they have a nucleus? The nucleus of a cell controls everything inside the cell. And we'll talk about that when we get into the cell. So we can either have an organism that has a nucleus or without a nucleus. We call it anucleic. If there's an A prefix in front of something, it means without. So without a nucleus, we refer to it as a prokaryote, and things with a nucleus we refer to as a eukaryote. We could also distinguish if it's one cell or many cells. We either say unicellular or multicellular. And the type of metabolism we could also classify it as. We could have autotrophs versus heterotrophs. Autotrophs make their own food by way of something called photosynthesis or chemosynthesis. And heterotrophs, like ourselves, we can't make our own food. We have to eat it, get it from our surrounding areas. So the domain eukarya is broken down into these 4. Protista, one-celled organisms, or they can be simple multicellular organisms. But they do have a nucleus, and this is where our molds and protozoa are, okay? Algae. The animal ones are always multicellular. Eukaryotic, they have a nucleus, and they don't make their own food. The fungi are eukaryotic, they decompose. Molds, yeast, mushrooms are all in there. And plant, they're photosynthetic, meaning that they make their own food. They're multicellular and eukaryotic, having a nucleus. Okay? Questions on that? So when we look at classification of humans, we break it down into further. And you should know these in order. Not so much as this, but this you should know. The domain goes down to kingdom, phylum, class, order, family, genus, species. When we talk about Homo sapiens, we talk about the genus species. This is becoming more and more specific. When we go up this way, it's less specific. If it's in the Animalia kingdom, we're talking about we're in the same boat as bears, and we're in the same boat as fish, and reptiles, and so forth. But for instance, where we are with classes, it's just things that have hair, mammillary glands, or blubber, like whales, right? But bears don't fall into that. Oh, no, not bears. Fish don't fall into that category, right? So we get more and more specific as we go down here. You should know these in order. Something I used when I first learned this in 7th grade was not so much about the D, but King Philip can order 5 great subs. It's something I came up with. There's probably tons of mnemonics out there, but you should know them in order. Okay. So living things are grouped according to characteristics. The smallest unit of classification is the species, the last part there. Okay? So similar physical and functional characteristics will fall into here. And when you get to the species, they can interbreed. The second smallest is the genus. All right? All living human beings belong to the same genus and species, Homo sapiens. Okay? So the proper way to write genus species, the notation over here, for example, Homo sapiens, this is really for the people who are going further with biology, so you write things correctly. The first letter of genus word is always capitalized. The first letter of the species is never capitalized. If it's typed, then both words are italicized. But if they're not typed, then you can't write italicized, so you underline it. Okay? So that's how you do that. It's pretty straightforward. So defining features of humans, we're bipeds, meaning that we walk upright with 2 feet. That makes us more superior than other animals. Even gorillas have problems. They have to go like this and then go, and back down like this. Why it's advanced that we're walking on 2 feet is that allows our hands to be free. We could be carrying weapons and still walking and still being able to knock down that dinosaur if we need to. Opposable thumbs. That's making your thumb touch your pinky. We're the only species that do that. Monkeys can't do that. Monkeys can do this, but they can't do this. This is an advancement because that allows us to create different instruments to hold and inform a new form of communication. Writing. Or being able to hold that club much stronger than the apes would. So we could hold the club much stronger and knock down that dinosaur. Monkeys can't do that. They can do this. I go to the Franklin Zoo in Boston. I go to the monkeys and go, I think they throw me a banana peel. So that's what makes it, that's an opposable thumb. Large brain, self-explanatory, right? And then capacity for language, all right, both spoken and written. So this is just kind of showing you the chimpanzee, how we hold people. It's difficult for them to wrap that thumb closer to their pinky. They questions so far? Pretty straightforward, right? Okay. Okay. All right, so let's talk about scientific method. All right? Science. Who decides what constitutes knowledge? Is there any difference between fact, or law, and hypothesis? What does it mean to systemically ask and answer questions? Is there an organization that you should be asking to make sure you cover everything? Can you generate explanations from natural phenomena that are consistent with current knowledge? The sun is going to shine tomorrow. It's going to rise because that's what's happened for the past eons. But there's a reason for that. You need to constantly revisit explanations to accommodate new knowledge. You need to go back to certain things. Is Pluto a planet or not? That changed things, right? So the scientific method is an organized description of the steps to make sense of something, all right? Helps us to develop a logical explanation of why something is happening. Utilizes both inductive and deductive reasoning. Rule out and rule in certain things. All right, so this is what we got to do. We got to think about this. We observe. You identify a problem by your observation. And you record your observations by collecting data. For example, you see your dog scratching. You're observing that. The dog is scratching. So your dog, Prince, we'll call him, is scratching himself. You're identifying a problem and you want to try to explain why he's scratching himself. Once you can explain this, you can brainstorm how to fix it. Well, let's use the scientific method to see if you can logically put this all together. Explain why Prince is scratching himself. Okay, so first step, you observe a problem. You want to try and do something. So you form a hypothesis, some kind of a tentative answer to a well-framed question. By using some previously explained knowledge, you form a question. You wonder if Prince has some type of infection or if something is eating or biting him. Maybe that's why he's scratching himself. You consult your pet manual and you form a hypothesis that he may have fleas. Okay, so you're picking up other information. Fleas. I never heard about fleas, but they seem to be biting and that causes scratching. Okay, maybe Prince has fleas. So we've got to go to step 3 now. We form an hypothesis. If he has fleas, he's scratching. Okay. So now you got to develop a prediction. And it should follow a logical reasoning from this hypothesis that you came up. You read some more about the subject and the consequences of fleas living on Prince. You try to put your thoughts into a logical format here. If it is true that Prince has fleas, then what would be logically to follow? Would treatments designed to kill or repel fleas bring relief for Prince? All right? Coming up with a prediction. Now you've got to test the prediction. A hypothesis has to be testable. So you execute a number of experiments. You might try giving Prince a control bath. Control with that. Well, you're just going to be washing him with soap and water. Nothing else. If that makes his itch go away, and you didn't put any kind of itching shampoo in there, you just did soap and water, if the itch goes away, then it's probably just dirty. You didn't have fleas. You should also do some research to determine if fleas drown in normal bath, right? If he still scratches constantly after his bath, you can try flea shampoo or a flea collar. If one of these works, then you might decide your hypothesis was the correct explanation of Prince's discomfort. Okay? One step. I know this takes longer than, you probably could figure this all out on yourself, but we're going step by step so you understand what the scientific method is all about. I'd rather use an example that's easy that all of you can follow. So now you've got to be able to repeat your test. A test has to be repeatable. So it has to be retested and retested. It should be reliable. Is your explanation reliable? Can it, if you do it again, you should get the same results. Can you make a generalization? Repeat the test next time when Prince starts scratching in the same way. Ideally, you would ask someone else to repeat your test. Maybe you're putting the shampoo on a different way because you see where he scratches, you're going to spend more time putting more on there. You're biased. It'd be better to have someone else put the soap on so they're not as biased. If Prince brings Princess home for a visit one day, okay, and you notice that she is also scratching, you might give her master, her owner, the same flea shampoo and see if it works. If Princess gets relief from her flea shampoo back, you may be on the way of making a general statement about the dog's scratching reason. So you've got to record all your results, the scientist would. You make a conclusion and document your successful and also unsuccessful experiments. It's okay to be unsuccessful. Just going back to the drawing board, but it's okay because it didn't work out. It makes you want to see, well, we've got to remove something, we've got to do the test a little bit different. You share your results with others and they'll add their results to yours. We'll be doing that in the lab room over here. Putting other people's labs, when we put them all, you'll do certain things and you'll see maybe for some reason you didn't wear gloves and you contaminated something. And that's okay. I'm not saying anything wrong with that. You know where you went wrong. And that's the important thing. But maybe the other group did it correctly. And we'll compare different things. You may have enough data to conclude that fleas are a reasonable explanation for the scratching problem and the active ingredients in the flea shampoo were the likely solution. So the summary steps, you identify a problem by observing, you make a hypothesis, an educated guess of what would happen, you make a prediction, you need to test and retest that prediction, repeat the experiment, and then make some sort of conclusion. Okay? Questions on that, right? Good example? Everybody can relate to, right? Okay. Now, hypothesis versus theory versus law. They don't mean the same thing. A theory and a hypothesis is not the same thing. Law is not the same thing. They're just three different animals, really. A hypothesis is a reasonable guess of what you observe. But you're observing a particular thing, not a general thing. For instance, when you looked at Prince, you're looking at one particular thing. Is it fleas that's doing this? That's causing him to scratch? Right? You're not looking at the whole thing on all the animals and stuff, because it's different. You're looking at more specific. So an example is that female birds prefer to mate with male birds that have longer tails. It's very specific. That's a hypothesis. A theory is when more than one hypothesis have been retested and proven, it may partially be true if later on more discoveries have happened. And then you build on it. I mean, Newton back in, I don't know what century it was, the 16th century, an apple falls on his head and says, oh, gravity did that. Or not gravity, but things will just fall. He came up with that idea. And that theory that if you let something go, it will go down to the ground. But we built on that. We learned about gravity. We learned about things that are being attracted to something. We built on that. So it may be partially true, but we learn more about it from other people's experiments or observations. Theories deal with a bigger, more generalized issue than what a hypothesis does. For instance, modern organisms descended from pre-existing life forms. A more generalized thing. Does that make sense? We didn't look at one particular thing, like the birds, how they mate and stuff. We looked at a generalized thing. Then we have something that's a law or a fact. This is usually short and straight to the point. Law of gravity. Boom. You know what I'm saying? It's just things will fall, will continue to fall. It's universally and mathematically being always true and can never be disproven. Example, living things are made up of cells. We know they're made up of cells. There's no question about that. See the difference here? So let's just do another example so you understand the three things. Let's use evolution as an example here. Evolution happened. It's a law. It's a fact. Evolution has happened. Okay? It's the law of nature. However, it does not describe the how and why it occurred. Okay? Describing the how and why it happened, this would be a theory. A good example is what we talked about before. Natural selection is a theory. It provides a host of descriptions for various mechanisms, describes the method by which evolution works. Okay? Describing one particular observation, that's a hypothesis. If seals stay away from polar bears, they will avoid being eaten. More specific, part of the natural selection, but it's more specific. Does that make sense? Okay? Questions about these three terms. You should know the difference between these three. Okay, some further thoughts. Inductive reasoning. Make generalizations based on observations about the world. Make logical explanations from the specific to the general. An example of this would be an observation. Every winter in the past was colder than the preceding summer. Generalization. Winter will always be colder than summer. Does that make sense? Right? Right? Then we also have deductive reasoning. Use general premises to make specific predictions. If organisms are made of cells, premise number one, and humans are organisms, premise number two, then humans are composed of cells. Deductive reasoning. Make sense? Okay. Some other things. Variables. We'll use the word variables. They're factors. Factors that vary in an experiment. They vary. They're factors that we can, temperature, pressure, we can change those variables. All right? A controlled experiment is an experiment that cancels out effects of certain factors by using what we call controlled groups. Two groups are always used. But one of those groups, we're not adding something to it. For instance, we did the Prince thing, right? We did the dog. A controlled group is that we're going to wash Prince, the dog, with just soap and water. We're not adding the ingredient. So one group is not being treated with a certain factor. It's left alone. So we're taking prints, we're washing with just soap and water. That's the control. Then we can take prints another time and do soap and water with the flea shampoo. So you've got to have a control group. If he doesn't scratch with the control group and he doesn't scratch with fleas or with the flea shampoo, the control group, that proves that it's probably not the fleas, unless they get drowned, right? Independent variable, a factor that affects the experiment. Okay, something you're doing to the experiment. In a perfect example, there should only be one independent variable. Kind of difficult to do, and I'll explain why. A dependent variable is how it responds. It has to be measured. But it's what the independent variable does. So an independent variable is what I change, what I can do to the experiment. A dependent variable is what I see happens, the results. So this is the best way I can explain it. You take water and you're going to pour it on the grass. The water itself, the sprinkling of water on the grass, I have control. It's independent. No one's telling me what to do. I'm controlling that. And I'm sprinkling on the grass. The grass is going to grow, or hopefully. It's dependent on the amount of water that I'm giving it. And I can measure the length of the grass. Does that make sense? So understand the difference between a dependent variable, depends, I hate using the word in the definition, but it depends or relies on the independent variable, what you do to the experiment. What I'm doing to the experiment is independent. I'm doing that. Okay? Does that make sense to everybody? A lot of confusion when they get independent and dependent variables in students. Okay. So just understanding cause and effect. The cause is putting water onto the grass, that's the independent variable. The effect of the grass growing or not growing is the dependent variable. Okay, and the last thing I just want to talk about, the scientific method is not perfect. So we have some issues with it, and these are just some that you should be able to understand. First off, it's been invented, that scientific method is invented by historians and philosophers, not scientists. We've got to think it's been based off of philosophy, all right, not the people doing the experiments. It infers that it must follow specific order, not necessarily. Okay? The test might have been an observation already, and it went out of order. It infers there is only one method, the scientific method. No, there's other methods that you could have done. I'm sure you had a dog that had the same thing, that was scratching, and you didn't use the scientific method. You used some other method that your brain created. The discovery of science, or discovery science, has more important contributions with very little dependence on the so-called scientific method. In other words, we discovered so many different things without even doing the scientific method. Even before the scientific method came out, we weren't even doing that. The bottom line that I want to just say, do an experiment by way of the scientific method is good. But just know that there's other ways that work, or just as good, or even better. but you should know the scientific method, because it's a very rational way of doing something to make sure that you cover it. Okay? Sound good?